Hey, I'm Mr. Williamson, and this video is about the cell cycle and chromosomes. For those of you that are new to the class, strong suggestion you take good notes. Again, I like Cornell style, but you can do whatever you want to do. Whatever makes your motorcycle go, it works for me. We will be turning them in the classroom. We'll discuss that during class, or we already have if everything goes like I think it should. But make sure you take good notes because they're going to be part of this assignment. Let's get started. Just a real quick review. We're talking about DNA. We're talking about getting the DNA from the mitosis from one cell to another after it's been replicated. Like you started out as one fertilized cell, one fertilized egg, and we've replicated the cell enough times so we're your 50 trillion, 37 to 50 trillion cells. All of them with the same DNA, hopefully. How do we do that? How do we get the DNA to replicate, which we talked about already in Unit 5, but how do we get it into the other cells? That's what we're looking at today. All right, so the quick review. DNA has a backbone of alternating sugars and phosphates. While they're not on this diagram, they're there. Hopefully that's review. And then this little guy right there, he's pointing out to that we pair up A's with T's and G's with C's. Straight line, crooked line, crooked line. Hopefully, again, that's a review. Whole purpose of DNA is to hold the code to make proteins. We start out with transcription up here. Transcription is copying the DNA over to messenger RNA. A's are copied over to U's, T's over to A's, G's to C's, C's to G's. Remember, there's no thymine in messenger RNA. There's only uracil. The messenger RNA travels outside of the nucleus because the DNA can't and goes to the ribosome, where transfer RNA, tRNA, brings in the correct amino acid based on the codons. The codons, again, are sequences of 3 bases in the messenger RNA. And hopefully you remember how to read the table on how to read a codon and interpret the correct amino acid. That should be a review of the last unit. We're talking today about the cell cycle. This is I like this. This is a really good image of the cell cycle. The thing about the cell cycle is that we can start anywhere. It is a cycle. So no matter where you start, you're going to end up back in the same place. I'm going to start right up here, right after the cell is divided. All right, I think it makes sense. Think of the cell cycle kind of like a life cycle, like your life cycle. We don't go in a full circle, but we do start out as a newborn, an infant, and then you go into like a toddler, and then a child, maybe a teenager, young adult, old fart, dead. That's your cells. That's your life cycle. Well, in this case, and I'm not to dead yet, so don't even think that. Yeah, we're going to start right here. The cell is divided, and we're now with one cell. This guy right there, one cell. All right. The phase that it's going to go through is called interphase. It's right here. Interphase. All right. It is not a phase of mitosis. It is a phase of the cell life cycle, but we're going to talk about it like it is a phase of mitosis because we're going to talk about the cell life cycle and include all the other phases. But right now, interphase is everything that happens when the cell is not dividing. An interphase can be divided up into 3 sections or 3 phases. All right, and we're going to talk about each one of those. But this is the cell cycle. You're probably going to want to come back to that because I think there's a worksheet that is involved with this assignment that you're going to need to fill out and submit for credit. But this is the cell life cycle. All right, it's 3 phases: G1, S, and G2. We'll talk about those. All right, G1 is where the cell does its normal cell thing. So if we were talking about a cell in your body, we'd be talking about a liver cell being a liver cell. It does its liver cell thing. Or a muscle cell doing its muscle cell thing. Or a brain cell doing its, oh, never mind. You've never experienced that. So, but it would be like doing a brain cell thing if you know, like they were working. Just kidding. They're working. My point is that G1 phase is really just the cell doing its cell thing. No matter what kind of cell it is, if it's in G1, it's just doing the cell thing. a signal comes along, and there's actually an enzyme that signals to the cell that we're going to divide. You're going to divide. A little message comes into the cell, probably through the receptor proteins that are in the cell membrane. Remember those, the receptor proteins? You got the cell membrane that, has phospholipids, and we just draw the phospholipids like this. And then you have a receptor protein that kind of goes through the whole thing. Remember the cell membrane? That was like unit 3. And then a message comes in and signals that, which sends out a message under the inside of the cell saying, hey, go do this. And then an enzyme goes to the DNA and says, hey, we're going to divide, and it creates the enzymes that are needed for division. Well, one of the things, the first thing that happens is the S phase. And the S phase is synthesis. We're going to replicate the DNA. In order to divide, we have to replicate the DNA. All right? All right? So we're going to replicate the DNA. Now, again, this should be review. DNA replication works. We unzip the DNA using DNA helicase. DNA polymerase comes in and matches the A's and T's, G's and C's. We're not going to use, because we're not making messenger RNA, we're making DNA, so the A's and T's, G's and C's all correspond like they're supposed to. This should look familiar, seeing as I took it from one of the previous videos. But you can see where you have the helicase coming in here, unzipping the DNA up that way. You have DNA polymerase, and this one's working this way. This one will be working this way. Remember that 3-5 anti-parallel part of the DNA, and looking at the backbones, the sugars are anti-parallel. They point different directions. Again, it's not like that's part of the test, but I'm trying to refresh your memory, because when I talk about it, it should sound familiar. So we're talking about the S phase in interphase, or the cell life cycle, and we're talking about DNA replication. The next phase is the G2 phase. The G2 phase is where the cell prepares to divide. Things that are needed for division take place. For instance, the cell grows, because we're about to split it in half, so the cell actually gets a little bit bigger. One of the things that we bring in is centrioles, especially in animal cells. Centrioles are responsible for producing spindle fibers. So we have, they look like this, all right? You have the centrioles that form in the centrosome. The centrosome is just a body of centrioles, and centrioles will go to the opposite poles, and we'll talk about that and what they do when we talk about mitosis later. But that's what the G2 phase is, bringing in the things that are needed for, there's a truck outside. Isn't that lovely? Centrioles are responsible for the spindle fibers. The next phase is mitosis. The actual cell actually divides. And we're going to talk about that in a whole different video. That's the next video. This whole phase right here, which is not interphase. It is not interphase. It's part of the cell cycle, but it is not interphase. It's the mitotic phases. There, you don't need to know this, because it'll be in the next video, but it's going to be prophase, metaphase, anaphase, and telophase. Don't need to know that right now. You will eventually, but you don't need to know it. What we're talking about are these parts right here. Those parts right there. The G1, S, and G2. We need to know those, what happens in each one of those. G1, the cell grows, it develops, and it does whatever its cell thing is. S phase is where we replicate the DNA, and then the G2 phase is where we get everything else in the cell ready to go for division. Okay? We're going to talk about diploid cells, or the diploid. I talked about that in the introduction to the unit, diploid and haploid, and the N number. Okay, and you will see a cell, like organisms, we are 23. Okay, if I could do this right, let's try that again. 23N. Okay, 23N. That means we have 46 chromosomes, 23 pairs of chromosomes. All right, there are, like corn. Corn is 90, I think it's 90. That's the number of pairs of chromosomes that a corn plant has. That means it has 192 chromosomes. All right. When we talk about the N number, we're talking about a diploid cell. So it's like our N number is 23. All right. But what we're talking about is this is a 2N plant. 2N times is the number of pairs of chromosomes. We're 23. So we would be 2n, twice as number of 23. After we've replicated the DNA, the cell becomes 4n because we've replicated it. It has twice as much DNA as it used to have. So the n number is the number of pairs of chromosomes. Again, we have 23 pairs. All right. So then when the cell divides, mitosis, which we're going to talk about in the next video, we start going back to being 2n, which is actually 2n and 2n. All right, and then we get back down here, it's 2n, and this one also is 2n. Now, if we were talking about haploid cells, sex cells, which I explained in the slightly in the introduction to biology pair deck, then we would be talking about n. All right, 1n. Why 1n? Because when you add 1n and another n, you get 2n. All right, 2n is a normal body cell. So the n number is basically when you see a 2n or a 4n or just an n, you're talking about the number of pairs of chromosomes in that organism. And not all organisms are different, but there is no regulation on the number of pairs of chromosomes in an organism. Fish might have one number, plants have another one. The number does not go up according to the complexity of the plant or the organism, because it's just a number of pairs of chromosomes, the way it works out. What I need you to remember from this is that when you see 2n, you're talking about a body cell, a cell that is not divided. If you see 4n, then it has to be a cell that's going to divide soon, because 4n does not exist and function as a cell because there's too much DNA. When you see 1n, then it's a cell that is a sperm or an egg, a sex cell. That is going to be combined with another 1n cell, and it's going to be 2n. Normal body cells in every organism is 2n. All right. There is a link between the replication and the chromosome duplication. We're gonna talk about like I'm going to show you DNA or chromosomes that look like this. What I need you to understand is that if you see a chromosome that's like this, and I will be drawing it kind of like X's. Just kind of what it looks like to me. When you see the chromosome like that, then you're seeing a already replicated strand of DNA. It's been through the S phase, because we start here in the unreplicated DNA. We replicate it, A's to T's, G's to C's, unzip it with helicase, etc. And we are now putting the similar DNA together. We are taking that DNA, this DNA and this one over here, DNA in this here is the same as the DNA in that one there. And what we need to do is figure out how to divide that up. All right? So let's look at chromosomes. Chromosomes, what we're starting with is a strand of DNA, unraveled. Just a long strand, 2 backbones, anti-parallel, complementary, T's with G's, T's with A's with, got that wrong, T's with A's, G's with C's, etc., etc. We're going to coil that around these proteins, all right? These are called histones. Wow, that's spelling. I think it's this, but I could be wrong. I don't really care. You don't need to know the name of it. I'm just telling you, they're called histones. Basically, they're just proteins that we coil the DNA. Think of it like a spool of thread. We put the thread around the spool because holding a whole blob of, a tangle of thread makes it hard to work with. But if we wrap it around the spool, I couldn't think of the word, the spool, then you end up with a neat, organized way of keeping that thread together. Well, that's what histones do. They wrap around these proteins. We wrap around DNA around the histone, and then we take that, we coil it up even more. So you can see we've taken the DNA here, and we've wrapped it around each of the histones. Then we take that and we coil it up all like into a spiral. All right, and we take all of that and we coil it up into the chromosome. So basically what a chromosome is a very highly organized form of the DNA and protein. Chromosomes are mostly DNA and protein. That's mostly DNA and a little bit of protein. If we see it in an organized form, it's going to be kind of like this over here. It's unraveled. We have to be able to get into it to unzip the DNA, to copy the messenger RNA, things like that. So we talk about protein synthesis. But when the cell's dividing, you're going to see it organized in a chromosome where it's tightly bundled and it's easy to work with. Because what we can do is split this chromosome in half, pull one half this way, one half that way, and we can now split the DNA and put it into two different cells. So going back to this slide, you can see where what I'm trying to do is take this chromosome and split it down the middle, take this one going that way and that one going that way, and we end up with the two different chromosomes in two different cells. If we organize it, it makes it easier to do. When we take, like in our body, when we take our chromosomes, we have 23 pairs of chromosomes, or 48 chromosomes. 46 chromosomes. Something to start thinking about. You get half of your DNA from your mom and you get half of your DNA from your dad. This is called a karyotype. A karyotype is a range picture of chromosomes at their most condensed state. That's okay. Really what it is, it's an image of the chromosomes arranged by size. We're going to be talking a lot about this when we get into the genetics and we get into and even into a little bit of evolution. But they're arranged by size. The biggest over up there, the smallest down here in 22. All right. And what we're looking for is similar shapes and sizes. And you can see where the coloring of this, by the way, is done artificially. I should probably find you a real, a much better picture. That would have been a better option. But this is the one I've been using for a long time. Oh, well. But you can see where not only is it arranged by size, but also where the centromere, and we'll talk about the centromere later, but the centromere is that little, where the X crosses. That's called a centromere. Talk about that in the next video. Don't worry about it. But some of them cross in the middle. Some of them cross at the top. Okay, some of them don't even cross. They just kind of come up to the centromere and they come down, like in number 22. That's a karyotype. Notice that all of them come as homologous pairs. Homo meaning the same. Homologous meaning that they're the same pairs. We have almost all the same. There is a difference down here, though. All right? And that question is that a boy or a girl? Well, the fact that it has an X and a Y, that equals a boy. If it has an X and an X, that equals a girl. You get one from mom and you get one from dad. A little bit of thinking and you can figure out which gender determines the gender of the baby. Does mom determine the gender or does dad? Let you think about that. We'll cover it later. But this is a karyotype. It's showing you all the chromosomes. Each one of those chromosomes is DNA wrapped around histones, coiled up and condensed into what we're making into a chromosome. All right? So in the next video, we're going to talk about mitosis, where we split up the chromosomes, and then cytokinesis, which is kind of like part of mitosis, where we split up the cytoplasm. One thing that can happen that when the cell cycle goes wrong is we end up with something like cancer. And this is an image, an electron micrograph image of prostate cancer, where it's started to run away. You can't tell looking at it. I'm just showing you this is actually cells dividing, just finishing up dividing. But when the cell cycle goes wrong, we can end up with something bad like cancer. All right, so that's the cell cycle and chromosomes. So real quick review. The cell cycle is divided up into two parts, the mitotic part, which we talk about next unit, and the interphase part, which is everything except mitosis. All right. Interphase can be broken into three sections. G1, where the cell is growing and does its normal thing. S phase, where it replicates. And the G2 phase, where the cell gets ready to divide by adding centrioles and growing bigger and things like that. All right. Then we talked about chromosomes, where we take the DNA and we coil it around proteins called histones. Again, you don't even know the name of it. Just understand that a chromosome is a lot of DNA wrapped around proteins, okay? Wrapped around the histones, and we coil that up, and basically we're getting the DNA condensed and organized and ready to divide in an orderly manner, which we do during mitosis. If you have any questions, make sure you ask me in class or in the Edpuzzle. Hopefully you took good notes. Good luck.